Hello, everyone, and welcome to our live webinar on the latest COVID-19 research. My name is Maria, and I'll be the moderator today. First, we'll have a 40-minute presentation by our guest, and afterwards, we'll have a Q&A. If you have any questions, you can type them into the Q&A panel to the right, and we'll answer them afterwards. Today's webinar is presented by Dr. Judd Hultquist from Northwestern University in Chicago. He has been involved with infectious disease research since 2008, especially focusing on HIV. He recently directed his attention towards COVID-19, where he's trying to identify different types of the virus. Thank you again for coming today, and Judd, please go ahead and start your presentation.
Fantastic. Thank you so much, Maria. And good morning, everybody. So I first want to start out by saying I really appreciate this invitation to present today. As scientists, sharing our work is such an important part of our job. And given current circumstances, it's been increasingly difficult to find sort of venues to share our work. So I really do appreciate this opportunity. I usually finish my coffee right about now. It's about 10:30 in Chicago. And so I'm not exaggerating to say I'm really excited to share our stuff with you today. So I guess today what I want to talk to you about is SARS-CoV-2 viral diversity, causes, consequences, and opportunities. This is really inspired by all of the recent stories that have been appearing in the media about SARS-CoV-2 mutations. So SARS-CoV-2 equals scary and mutation equals scary. So SARS-CoV-2 mutations equals very scary, which is really made for good headlines. But the reality, of course, is a lot more complicated than that. So most mutations are in fact harmless and serve as a remarkable tool for us as researchers to track viral spread and inform public health policy. So today I want to take a deeper dive with you on the causes and consequences of SARS-CoV-2 viral diversity and to look at some of the opportunities that this diversity affords us. So the first thing to understand is that all viruses mutate or change over time. Mutations can be sorted into two basic categories: intrinsic mutations and extrinsic mutations. So intrinsic mutations arise due to internal properties of the virus, such as the fidelity of the polymerase and its ability to proofread and or correct its sort of mistakes. Now, extrinsic mutations, on the other hand, arise due to the influence of the cellular environment. So that includes NTP imbalances, and the presence of RNA or DNA editing enzymes such as the ADAR family or the ApoBec family of proteins, respectively. Now, both of these factors, intrinsic and extrinsic together, define what becomes the viral mutation rate. Now, the second thing to understand is that different viruses mutate at different rates and at relatively constant rates. So as shown in this chart here on the left, we're comparing genome size on the x-axis to mutation rate on the y. Generally, the longer the viral genome, the lower the mutation rate. And this holds true all the way from eukaryotes like us with very long DNA genomes through prokaryotes or bacteria and into the virus families. So RNA viruses in particular have some of the highest known mutation rates recorded. HIV and influenza viruses, for example, have notoriously high mutation rates that have resulted in significant problems in vaccine design and in antiviral resistance. So just as one example, because HIV is my favorite, HIV makes an error in 1 of every 5 genomes produced on average. So in an infected person who has not achieved viral suppression, as many as 10 billion to 1 trillion new viruses can be made each day. So that means the virus can explore an absolutely incredible amount of mutational space, even within a single individual in a single day. Now, if these mutations fall into drug or antibody binding sites, they may result in resistance mutations, which can render treatments ineffective. And this is what's illustrated in this cartoon on the right. So if we have an initial infection event shown here, over time, the virus will diversify. And if a drug treatment is started and an antiviral resistance mutation is present in the population, you'll very quickly get selection and the evolution of an antiviral resistant population, rendering the drug ineffective. Now, if this drug is stopped, of course, the population can continue to diversify once again. So besides contributing to antiviral resistance and immune escape, some viral mutations have also been observed to influence spread, and that both is spread between species and spread between people. So just as an example, again, looking at HIV, it required a number of mutations to effectively jump from monkey populations into the human population. These mutations were required to overcome the human immune system and to optimize replication in a new host. Similarly, during the Ebola virus outbreak of 2014 to 2016, a number of mutations were cataloged, some of which influenced the transmission from one person to the next. Coronaviruses, like SARS-CoV-2, also required mutations to jump from their host species, which was originally bats, into humans. Now, it's thought that SARS-CoV, the original outbreak in 2003, was caused due to a transmission event from bats to civets, civets to humans. MERS coronavirus, on the other hand, went through camels as an intermediary, and it's thought that SARS-CoV-2 probably came through pangolins, though that is still under active investigation. And of course, now that it's in the human population, it's continuing to mutate even further. So over time, we're seeing SARS-CoV-2 diversifying both across the globe and over time. This dendrogram is showing just the diversity cataloged in the open access GIS aid database. And this is just showing diversity between December of 2019 and May of 2020. And of course, that continues to progress today. Now, luckily for us, SARS-CoV-2 has a much lower mutation rate than viruses like HIV or influenza. If we refer to our chart earlier comparing genome size and mutation rate, SARS-CoV-2 actually falls on sort of the extreme low end for mutation rates of RNA viruses. And this is primarily due to two things. So first, the SARS-CoV-2 genome is huge. It's among the biggest RNA viruses in humans that have been categorized to date. And as such, the polymerase is actually thought to be a little more accurate than most RNA viruses. But the second and major reason is that it also includes a proofreading protein called NSP14 that helps to correct errors made by the polymerase, lowering this mutation rate even further. So just to kind of summarize, SARS-CoV-2 is changing over time. It has a relatively low mutation rate, but that does not mean it's not continuing to diversify. So how can we use this to our advantage, and how can we determine which of these mutations might be harmful? And as it turns out, there is a lot that you can learn from just a few mutations. So while some mutations may be harmful as they contribute to antiviral resistance or they may improve transmission, these are really the exceptions to the rule. A vast majority of mutations that occur are neutral or are even deleterious to the virus, and they can be very helpful in informing researchers as to how, where, and when a pathogen might be spreading. So for example, based on how similar virus genomes are, we can determine what the ancestry of these viruses are. This is known as phylogenetics. Indeed, given the somewhat predictable nature of the mutation rate of viruses, that can be used as sort of a molecular clock to estimate rough timing events of when different viruses were introduced into different populations. Furthermore, by looking at the sequences, we can determine which of the mutations that are occurring were significant events along the evolution of the pathogen. And these can be looked at both structurally and functionally to determine which ones might contribute to antiviral resistance or transmission differences. And then finally, if spatial metadata is associated with the samples, the routes of transmission can also be inferred from the phylogenetic trees. So this type of research that I'm just illustrating here on the slide is being done across the world with different sequences that are being added to every day. And this is being used to inform vaccine strategy, public health practices, and clinical protocols. But to give you sort of an example from real life, I now want to go into sort of a deeper dive of what we're learning about the pandemic here in my current hometown of Chicago, Illinois. So just to get us all started on the same page, I wanted to make sure that we got a brief rundown of the pandemic in the US currently. So in this chart, which is available from the CDC, I'm showing case count and the blue bars with a rolling average indicated in the orange line. Now, the first case in the US was confirmed in Washington State at the end of January, January 20th, with a second case confirmed shortly thereafter here in Illinois on January 23rd. The first person-to-person transmission case that was confirmed on January 30th when the individual passed the virus to their partner here in Illinois. Later, about a month later, on March 1st, the first confirmed case was reported in New York State. Between March 21st and March 23rd, a whole series of states began to shut down. Illinois, New York, and Washington all issued stay-at-home orders within this tight window as infections began to rise throughout the US. The US exceeded 1,000 deaths on March 26th and 10,000 deaths shortly thereafter on April 6th. The US hit 1 million COVID cases at the end of April. But despite the fact that we kind of had achieved this plateau with our social distancing and public health measures, all US states began to enter reopening phases by May 20th. Shortly thereafter, the US exceeded 100,000 COVID-19 deaths, with 2 million cases shortly thereafter on June 11th. Since the reopening has occurred, we've been seeing a surge in cases, primarily in the South and West, but now this is beginning to be apparent here in the Midwest, and we're rapidly gone from 2 million cases to 3 million cases, and then 3 million cases to 4 million cases in only 2 weeks. So this is a brief rundown of the US COVID-19 pandemic as a whole, but it actually looks quite different when you look state to state. So deaths and cases in Illinois were actually relatively slow to rise compared to Washington State and New York. So as I had indicated on the last slide, the first reported cases in Illinois and Washington State were relatively at the same time. But in spite of that, Washington State's infections started rising relatively early as one of the first hotspots in the US. While in Illinois, we kind of got this slow petering of infections until cases rose a little bit later. Now, New York State, on the other hand, didn't report its first case for nearly a month after Illinois and Washington, but it saw an extremely rapid rise and a much higher rate of cases than Illinois and Washington. This is likely due to a whole number of issues, including public health measures that were present at the time, population densities, etc., demographics, community mitigation measures, the testing that was there, healthcare infrastructure, and the public health reporting. But the big question kind of was, is there a viral mutation that is also contributing to the differences that are being observed across the United States? So to get a better understanding of the viral mutations that were occurring here in Chicago, we adopted a whole genome sequencing strategy that's illustrated in this sort of line diagram here. So through a collaboration with Northwestern Memorial Hospital, we became a central repository for incoming diagnostic tests that were left over after patients would get tested. We took 127 of these nasopharyngeal swabs or brachioalveolar lavages and extracted RNA from those. The RNA was reverse transcribed into cDNA. The cDNA was PCR'd up to identify the SARS-CoV-2 amplicons. These were barcoded and multiplexed and subject to whole genome sequencing using the nanopore platform. Virus reads were then assembled, and at the end of the day, we arrived at 88 assembled genomes. The protocol we used was actually adopted from the Arctic Network's original protocol that was reported fairly early in the epidemic. We are achieving nice reads across the entire sequence. For us to consider a sequence whole, we required at least 10-fold coverage over 90% of the genome, and we were able to achieve median read depths of about 348 reads per amplicon. Because the genome is so large, you actually end up needing 200 roughly amplicons to cover the entire thing, each amplicon being an average of 400 base pairs long. So this was our strategy. From the 127 original sequence samples, we were able to get 88 complete genomes. We took all of these samples in while testing and infections were just beginning to ramp up here in Illinois. So this was during the third week in March, between March 14th and March 21st. About 50% of these were from the ED, although a quarter of them were from inpatients and roughly 10% were from patients in the ICU. So when we looked at these sequence and compared how similar they were to one another, we could get an initial phylogenetic tree. And when we looked at the phylogenetics, we quickly realized that there were roughly 3 predominant clades circulating in the Chicago area at this point in March. We just assigned them clade numbers based on the size of the clades at the time that we did the sequencing. So we have clade 1 shown here in blue, clade 2 shown here in purple, and clade 3 shown here in red. So for sort of phylogenetic experts out there, clade 1 corresponds to the pangolin labeling schema B.1, clade 2 corresponds to A.3, and 3 corresponds to A.1. So to better understand how these 3 clades relate to the broader epidemic in the United States, we compared these sequences to all of the available sequences available in the US at that time. And what we saw was actually quite interesting. So our clade 1 viruses were most closely related to viruses that we saw on the East Coast, particularly viruses that were circulating in the tri-state area of New York, Connecticut, and New Jersey. The clade 3 viruses, on the other hand, were very closely related to the viruses that were coming out of Washington state. And the clade 2 viruses, we saw almost nowhere else except for our original Illinois sequences. So we could take this information, and as I indicated earlier, use it to map it onto the geographical spread, but then also try to understand when these viruses were being introduced into our population. So going clade by clade, here we're just showing the geographic spread of clade 1 at this point in mid-March. And as you could see, and as was evident from the earlier phylogenetic tree, you could see that this clade was primarily centered on the East Coast and in New York, but then achieved relatively broad spread across the United States. Clade 3, on the other hand, was our West Coast clade. It was primarily centralized in Washington, but was also spread elsewhere, and it got some limited spread into the Midwest. And then clade 2 was our Chicago relatively specific strain. We saw some sequences in Wisconsin, a few in Georgia, but primarily this clade was made up of sequences from here in Illinois. When we estimated the rough timing of this, it appeared that these sequences originated in Illinois sometime in late January and percolated slowly, spreading eventually more broadly sometime in early March. And it was at that point that it also started spreading to some of these other US states. So using the same information, we can estimate the population size of each of these clades over time in Chicago. And what we were seeing was that clade 2 and clade 3, so this was the clade from Chicago and the clade from Washington, both were introduced at roughly similar times. But while clade 3 expanded in Washington state, clade 2 just continued to sort of percolate. Clade 1, which was introduced later into New York, of course, exploded very rapidly and became the predominant clade in the United States. So to understand this a bit further, we then took these sequences and put them together with the sequences available across the globe at that same point in time. So here we're doing the same phylogenetics, but looking at sequences available from across the globe. And what we found was that, similar to previous reports, that clade 1, which was our New York representative clade, actually was most closely related to sequences coming out of Europe. Clade 3, which was our Washington clade, was most similar to other sequences from the West Coast. But surprisingly, our clade 2 viruses were also very heavily represented primarily here in Illinois. So taking this sort of global approach, we can then again go back and look at the geography and the temporal dynamics. And what was really striking to us was that this clade 2, this Illinois clade, appeared to originally have been most closely related to the sequences from Wuhan, from the Wuhan province of China. So we had initial spread of this clade early. Eventually, we got propagation into Illinois sometime in January, where it continued to spread slightly and then expanded from there. The other slight surprise is when we looked at representation across the globe, we did see this clade also expanding in Australia, and we think this actually was a transmission event from China to Illinois to Australia. So just to quickly kind of sum up, we hypothesized that the Chicago-specific clade 2 viruses originated from the initial, roughly around the time that the initial reported cases were being reported, and these were coming more directly from China. Clade 1 viruses came east by the way of New York and Europe, and clade 3 viruses came from the west by way of Washington state. And relative to the other two clades, clade 2 viruses have spread relatively little, and especially in comparison to these clade 1 viruses. So the epidemic in Chicago, at least in mid-March, was broadly representative of the epidemic across the US and the global pandemic as a whole. So our major question at this point in time then was, is there a viral mutation responsible for the limited spread of clade 2 viruses relative to clade 1? And we were actually kind of fortunate that Chicago was this kind of melting pot of viral diversity, as we could compare more directly the clinical and demographic characteristics associated with each viral strain within a single hospital system. And this actually is really important because it controls for a lot of possible confounders regarding diagnostics, standards of care, and missing values that can plague very large clinical data sets that are compiled across several different hospital systems. So based on our demographic and clinical information that we were able to extract for each sequence, we fit a linear mixed effects model to these data to account for covariance to try to look for significant co-correlates of clade. And while we found that a lot of things were not significantly correlated with clade, including BMI, age, sex, race, ethnicity, source, severity group, isolation source, and isolation date, we did find that we had a significant association between CT value, or a measure of viral load, and the clade. So here we're just zooming in on that data specifically. And what we found was that clade 1 viruses, these viruses that were coming from New York, had lower overall CT values, which means higher viral loads, in the upper airways of our patients at the time of diagnosis. And this is compared to our clade 2, clade 3, or ill-defined clades that were occurring elsewhere in our hospital at the time. And this was pretty striking to us that these different viral genotypes were being associated with differences in viral load. The biggest question that we typically got from these initial reports were an explanation of timing. Could this be explained by the time that the sample was taken? And that's actually a really important question. So in an unrelated study, we're looking at viral loads in long-term hospitalized patients here at Northwestern Memorial. And what we're finding in these patients is that regardless of sort of their disease outcome, we're seeing this slow but steady decrease in viral load over time. And again, these are all hospitalized patients. So even if the disease severity remains high, we still see sort of these viral loads sort of tapering out globally over time. So if we were sampling later in the infection time course in one clade versus another, it could account for these differences in CT value. So in an attempt to control for that, we did case reviews to identify the time to symptom onset, as well as the time since exposure for our clade 1 versus our clade 2 viruses. And what we found is that there was no significant difference, either in the time to onset between clade 1 and clade 2, or in the time to exposure clade 1 to clade 2. Now, we couldn't get these numbers precisely for all of our patients, but in the patients we were able to get that data from, we don't see any significant difference. The second biggest question that we most commonly got is this occurring, everywhere, or is this only occurring in the upper airways? This is a question that we're still trying to address, but we do know right now, at least, that it's more obvious in the upper airways, so in sort of the nasopharyngeal samples. When we separate out our bowel or our lung lavage samples versus our NP samples, we see this very significant difference by clade in our nasopharyngeal swabs, but not necessarily our BAL. But of course, our BAL numbers are really low, so we're trying to bulk up our sample numbers to look at that more closely. One last thing I wanted to note from our clinical data was that viral clade does not appear to be associated with disease severity. We binned our patients based on mild, moderate, or severe disease, and we saw no significant association between either clade or CT value at the time of diagnosis. There are, of course, a lot more sophisticated ways to look at disease severity, and we're beginning to do that, and we're also beginning to look at specific symptoms. One of the nice things about all of this viral diversity being within one hospital system is we have standardized clinical records that we can dig into and correlate specific symptom recordings with each of these various severities and/or clades. So based on this information together, we hypothesized that these clade 1 viruses, these viruses that were originally in New York, are carrying a mutation that enhances viral transmission. And this has been borne out by later sequencing that we have done since March. So our original sequencing was all done again during this 1-week time period shown here in this temporal phylogenetic tree in March. But when we look at later sequences that we've isolated, we can see that our clade 3 sequences have completely disappeared. Clade 2, there are a few, but currently a predominant population of patients that we're seeing are all having clade 1 viruses, which would be consistent with a difference in viral transmission. So to begin to try to understand where these differences in viral load may be coming from, we were able to look back and see what mutations were present in each one of our clades. So here I'm just pulling out the clade defining mutations for clade 1, clade 2, and clade 3. And of course, the mutation that instantly pops out was this very intriguing mutation in the spike protein on the outer envelope of the virus, a D614G. Now, this mutation has been made famous by a number of different groups who have noticed that this particular mutation is becoming more and more prevalent over time. So this is an image from an excellent paper by Korber et al. That came out in Cell just recently, showing that this variation is originally in the epidemic, almost all of the sequences carried this D, which our clade 2 and clade 3 viruses carry. But as time goes on, this G614 variant is becoming more and more dominant, and that's the variant that we see in our clade 1. What was really kind of cool about this paper was that they also did some in vitro work with these viruses, introducing this614G mutation into the spike protein on pseudotyped viral particles. And what they were able to show was that you got more infection when you use the standard MOI just by including this one mutation in the spike protein. So the question, of course, is, does D614G alter viral transmissibility? What we know is that viruses carrying that mutation have higher viral loads in the upper airways. We know that viruses with this mutation have become predominant across the globe over time. And we know that viruses with that mutation infect cells more readily in vitro. And so probably is the best that we can come up with. Scientists tend to be very conservative in their predictions, and for good reasons. If it walks like a duck and swims like a duck and quacks like a duck, that means that we should definitely get an R01 to study it for at least another 5 years. What's really missing here for us to make the claim about transmissibility is we need a good transmission model. In order to really firmly determine the impact on transmissibility, we need those models, and a good one has not yet been developed, though the NIH is actively encouraging grants and people to begin to develop these kind of models, because they're really important questions and we'll need them to answer these important questions. So the D614G was one of the mutations that really stuck out with us and that several different groups are looking at, but there are other mutations in here that are pretty intriguing as well. So for example, the clade 3 mutations, which clade 3 again was in Washington State, there are a couple really intriguing mutations within the RNA binding site of the NSP13 helicase. So the virus encodes a helicase that's involved in viral replication. And these two mutations have the potential to disrupt the RNA binding site of that helicase, which could potentially alter the enzymatic activity of this protein. In work that we've just been beginning to do, we're seeing that these mutations do alter the protein steady state level, at least in vitro. What's also interesting about these mutations is they also map to the exact site as small molecule inhibitors that have been previously developed for MSP13. So if these inhibitors were to become effective treatments, this would be one potential place to look out for antiviral resistance. So to just kind of sum up what we talked about today, all viruses mutate and change over time, but most of these mutations are harmless. And in rare circumstances, they may influence viral pathogenicity. What's really important about this is we can use viral mutation to monitor disease epidemiology, and this can be a really powerful tool to inform public health strategy. So SARS-CoV-2 may have actually already evolved to transmit better in the human population, but again, until we have the animal transmission models, that's still going to remain a question that needs answering. So in the future, what we're beginning to do is we're looking at those longitudinal samples from hospitalized patients to try to understand if these viruses can evolve within a host, similar to how influenza and HIV can evolve within a host. We're also looking at intrahost diversity using variant sequencing by anatomical location. And we're using this technology to trace transmission events between patients and healthcare workers at our local hospitals. We're beginning to perform functional studies on the viral variants to determine their impact on molecular mechanism. And then we're gearing up to perform on-site epidemiological tracking across the city in collaborations with the University of Chicago, North Shore, and Lurie's Children's Hospital. So with that, I wanted to thank the really amazing team here at Northwestern, especially in the infectious diseases department. I want to particularly highlight the contributions of Ramon Redondo, who did a majority of the phylogenetic analysis. He's a computational wizard. We couldn't have done this without him, as well as Ian Ozer, who really helped with a ton with the deep sequencing and initial analyses. And Lacey Simmons, who's my lab manager and who has been organizing, these massive clinical databases for us. So if you want to learn more about anything that I talked about today with the Chicago, we do have this information, which is currently out for review up on medRxiv. We have several different funding sources. And again, thanks to STEMCELL for letting me present today.
Excellent. Thank you so much. If you could please pass me over the presenter mode, and then I'll take some questions from the audience. If anyone has any questions, you can type them into the Q&A panel, and I'll start with some questions we have here. Let's see. So first question, despite the virus spreading a lot in the US currently, fewer people are dying than earlier in the epidemic. Do you think the virus has mutated to make people less sick?
We can't really answer that question at this point in time. We do know that the demographics of who is getting sick has changed quite substantially from the initial wave to what we're seeing now during the surge. So you've probably seen some of the news reports indicating that younger people are now becoming infected, and younger people generally have better health outcomes. So it's probably just due to a difference in the demographics of who's getting ill, not necessarily a change to the virus itself. But again, that's a really difficult question to answer, and a lot more research would need to be done about it.
We have a second question here. Does this new mutation that seems to improve the virus's ability to spread make public health measures like social distancing less effective?
The short answer to that is no. So the public health measures that are in place, such as social distancing, wearing a mask, washing your hands, regardless of how quote unquote infectious on a pound for pound basis the virus is, all of those measures should be equally as effective. The mask should prevent transmission by spread through viral aerosol droplets. Washing hands, of course, removing it from your hands in case you're touching your face. The social distancing also to help with that droplet transmission. And regardless of how quote unquote transmissible a virus is, those measures should be effective. So if everybody could adhere to those, we could make a huge difference.
Definitely. We have another question here. Someone would like to know, what are the criteria between the mild, moderate, and severe categories of disease severity?
Yeah, so we base that based on time in the hospital, the need to be intubated. Of course, the worst outcomes and death were lumped into severe. And these details are all available in our medRxiv paper, but it generally was where they were located, so ICU, their need for intubation, as well as their ultimate outcome.
Got lots of good questions here. The next one is, generally speaking, how often do mutations affect vaccine development? Will multiple vaccines be required for different strains of the virus?
Yeah, so generally in vaccine design, breadth of coverage is a really, really important consideration. So when you're designing a vaccine, ultimately you want to cover as many different viruses or strains of the virus as possible. So when it comes to designing vaccines for SARS-CoV-2, they should have already been taking into account the mutations that have been present, including this mutation on spike. So ideally, this variation will not affect the efficacy of a vaccine, but again, that's going to be a vaccine to vaccine basis. And that's one of the huge struggles with coming up with a universal vaccine for influenza, for example. There's so many different subtypes and strains of it that having one universal and effective vaccine is just nearly impossible. Luckily, SARS-CoV-2 is not mutating nearly as fast as, for example, flu and HIV. So I hope that we can still come up with some, at least, protective vaccine for a length of time. That would be pretty broad, but we'll see. There's certainly a lot of people making them, so.
So this question is kind of along the same lines. Many antivirals have been developed for HIV. Why don't they work with COVID-19?
Yeah, so most of the antivirals designed for HIV work by inhibiting HIV enzymes that virus encodes. So if SARS-CoV-2 doesn't encode those enzymes or enzymes similar to it, those same drugs don't work. And that's the case here. So inhibiting viral enzymes can be a really effective way to make an antiviral, but then you need to have those specific inhibitors, and we just don't have those yet.
Unfortunately.
Yeah.
Do you have time for some more questions? We have a couple here.
Yeah, absolutely.
Okay, so this one is, have you explored the possibility that patients treated for cancer with radio and or chemotherapy might become incubators of viral mutants?
Short answer, no. I know there is a lot of interest to try to understand, especially in hospitalized settings where there might be immunocompromised people or people who are being actively immunosuppressed, for example, if they're getting some sort of organ transplant, if they could be carrying various viruses for a long period of time. But I'm not aware of any studies that have looked at that specifically.
Cool. Next question is, due to the recent surges in states like Florida, is it likely there'll be another mutation that could increase pathogenicity, or would it have the opposite effect because more people are being infected?
Yeah, so there's no particular selective pressure for pathogenicity. So for example, with the ability of this virus to spread potentially better, that would make sense because a virus, if it's able to spread better, you'd get more of it, and that's how natural selection, would work. We do have evidence that as this quote unquote clade 1 series of viruses continues to spread, it is continuing to diversify and form more clades off of itself. But none of those have been linked to any difference in disease severity.
So there's another question here. How does these mutations affect the different testing available? For PCR testing, are primary design affected by these mutations?
No, so fortunately, the PCR testing through the nasopharyngeal swabs, none of these mutations have been shown to alter our ability to detect the virus in those swabs or affect the applicability of those sort of assays themselves. What is kind of interesting to think about is if it turns out that certain strains do become more pathogenic or easier to transmit than others, we could develop high specificity PCR tests to look specifically for one type of virus or another. And that could be kind of used to do sort of risk stratifications in a clinical setting. Of course, right now, most of the viruses circulating are of this clade 1 type, so it's probably not particularly useful, but it would be as it continues, it's always an option.
Great. Another question here is, could these viral mutations have any influence on how diagnostic tests are run or distributed? Why did we repeat that?
Yeah, again, it's very similar to that last question, where if over time it turns out that a particular mutation is associated with a clinical outcome, you could develop a high specificity PCR test to specifically look for that outcome. That, of course, as I tried to convey today, is a lot more complicated maybe than initially you would hope, but it's definitely in the cards.
We have a couple more questions and then we'll end the session today. So this one is, from a point of view of viral mutation, what's your thought on the SARS-CoV symptom diversity? Why does someone have no symptoms while others are deadly symptoms?
Yeah, isn't it amazing? It really is one of the biggest mysteries about this virus, how a single etiologic agent can cause such a wide array of phenotypic disease outcomes. And what we think at this point is that it's probably most host related. We're seeing some diversity in the virus, but not nearly enough to account for all of the differences in disease outcome. We do see differences in patients' medical histories and exposure to different types of viruses, but again, SARS-CoV-2 is relatively new. Maybe if somebody got a related coronavirus, you might get some protection, cross protection against this coronavirus, but that's not definitive or shown at all either. So we think that mostly the differences we're seeing on a person-to-person basis are due to differences of that person themselves. So differences in their genes and differences in their immune system.
So lots of research still to be done here.
Oh yeah.
So last question here is, what's your opinion on monoclonal antibodies for treatment of the prevention of COVID-19?
As sort of a vaccine type strategy, coming up with one antibody that could neutralize all of them. It's definitely possible. As we've been seeing with this SARS-CoV-2 diversity, the viruses are diverse, but they're not mutating nearly at the rate of some of these RNA viruses that have historically been so challenging for us. Now, the bigger problem that might arise is our ability to induce sort of long-term protection. So it's one thing to identify a vaccine or an antibody that's going to be effective, but it's another thing entirely to have one that's going to be effective broadly over a long period of time. And that's kind of the challenging aspect to providing a vaccine currently.
So we're still getting a few questions here, so I'm going to read this next one. In terms of models for transmission, are these mathematical models or better cell-based models? Sorry, I get so many questions. You're a very popular guy right now, Judd. Let me see here. In terms of models for transmission, are these mathematical models or better cell-based models do you think are needed?
What we really need to do is develop, of course, developing better computational tools is always fantastic. But our sort of critical gap right now is developing a transmission model, particularly an animal model. So for example, I know a lot of people are looking at, can we develop mice that are susceptible to SARS-CoV-2? And then you can put infected mice with uninfected mice and look at the different parameters that influence transmission between them. And people are also looking at ferrets and hamsters as potential models. But until we get, a real hard data on which animals and which models are best to use for it. There are a lot of just sort of open questions.
Excellent. So thank you everyone for coming today and for sticking with us through our technical issues here. Thank you for coming, Judd, as well. It was a great presentation. We have a next live webinar coming up, and it's about the impact of SARS-CoV infection on the central nervous system. There will be an email sent out with a link to this webinar, and it's on August 5th. So everyone, please come and join us. And thank you again for coming. So have a good day.
Awesome. Thanks, Maria. Stay well, everyone.